The membrane that separates the cytosol and the outside of the cell is semi-permeable, which allows nutrients to get into the cell and wastes to leave the cell. It's important to understand that not all the molecules can cross the membrane directly. Molecules that can do it are usually small and hydrophobic. They can easily cross the hydrophobic inside portion of the membrane. There are several types of transport across the membrane. Passive transport requires no energy input. Molecules in passive transport move down their concentration gradient from the high concentration to the low concentration. The typical example of the passive transport is the diffusion. In diffusion, like shown on that image on the right, molecules of the dye move from their high concentration on the left of the beaker across the semi-permeable membrane to the low concentration area on the right side of the beaker. Now I have to make it very clear that any molecule can diffuse if the membrane is permeable for that molecule. Now imagine the situation when you have a beaker separated into two parts, and there's a lot of solute on one side and very little on another. Okay, so a lot high solute concentration corresponds to the low water concentration. Therefore, water will move from low to high concentration of solute. Water will basically try to equalize the solute concentrations on each side of the membrane. It will try to dilute the concentrated solution. We call this process of water diffusion towards the higher solute concentration osmosis. Osmosis is extremely important, the regulation of the cell shape and cell function. Normally, the concentration of solutes outside and inside of the cell are the same. We call such solutions isotonic. In isotonic solution, where concentrations outside and inside of the cell are equal, cell does not change its shape. When cell is placed in hypotonic solution, solution with a concentration of solutes lower than in the cell, water will start to flow towards higher solute concentration, meaning that water will flow into the cell. Cell will swell and may eventually burst. In the hypertonic solution, concentration of solutes is higher than in the cell. Therefore, fluid will move from the cell into the surrounding solution, and cell will start to shrivel. As I mentioned before, only hydrophobic molecules can cross the membrane directly. Transport of hydrophilic particles, such as ions, sugars, or amino acids, requires transmembrane protein, such as channel. These integral transmembrane proteins allow molecules to move across the membrane in either direction, and the direction of movement will entirely depend on the concentration gradient. So if the concentration of certain molecule, like on this image, is higher outside of the cell, it will move through the transport protein into the cell. Okay? Diffusion always happens down the concentration gradient, and it never requires any energy input. Unlike diffusion, in active transport, energy from the breakdown of ATP molecules is required to move the chemicals against their concentration gradient. in that same example with the slope, you need to apply energy to move the ball in this image up on the slope. So this image shows you the transport of, for example, sodium from the area with the low sodium concentration inside the cell to the area with a high sodium concentration outside. Okay? There's molecules, there's transmembrane molecules called pumps. Okay? They break down ATP into phosphate and ADP and use energy of that breakdown to shuffle sodium outside and bring potassium inside. There are many different pumps. Sodium-potassium pump is just one example of many molecules that can move various ions against their concentration gradient. Sodium-potassium pump is one of the most important pumps in the human body. It establishes the proper concentrations of sodium and potassium inside of the cell and outside of the cell. That establishment of concentrations of sodium and potassium allows the proper absorption of nutrients in the small intestine, for instance. Various pumps regulate the acidity of the stomach. They regulate the concentration of iodine in the thyroid gland, and so on. So, last topic that I want to touch upon in this particular video is transport of large molecules, such as proteins or maybe even cellular fragments. This transport is done by endocytosis and exocytosis. So this transport is always active transport. It always uses the energy of ATP breakdown. In endocytosis, shown here, the content that is outside of the cell, like this particles here, okay, these particles are engulfed by a structure called endosome. Okay, this structure is then fuses with a structure called lysosome. Lysosome contains acid and various enzymes that break down chemical molecules. So the content of endosome gets digested by the lysosomal enzymes and lysosomal acid. Think of it this way. Endosome is some kind of a cellular mouth, quote unquote. This mouth captures the particles, takes it into the cell, where this endosomal content fuses with the lysosome, cellular stomach, and the content of the lysosome digests the content of the endosome, releasing chemicals into the cell. When cell needs to secrete something, it uses exocytosis. Okay, here you can see secretory vesicle, which reaches the cell membrane, fuses with the cell membrane, and releases its content outside the cell.